Hi, now let's discuss about vaccine and viral infection and also requirement for effective vaccine development. So what are vaccines? Let's first take a look at a girl who are infected with a smallpox. And in this picture was taken in 1973, and you can see a rash throughout all the skins. And this has about mortality rate of about 1/3 of these people. So vaccines are very important. And what are vaccines? Vaccine is a biological substance to provide active acquired immunity to a particular infectious disease such as the smallpox. And vaccines can be produced to treat infectious disease that are caused by viruses or bacteria or parasites. And these vaccine is usually used as a preventive measure. We call it as a prophylaxis. So prophylaxis means a prevention. So action taken to prevent the disease. And also vaccine could be useful for treatment of some types of cancer. So in history, you remember Edward Jenner was the first one to develop a vaccine, and that changed the world because we now have immunization. So these smallpox in the history in Aztec, 16th century, and there's a drawing of these victims of smallpox. You look at this is very similar. And at the time, it actually swiped a lot of population because of this smallpox. So then we discuss about the virus and how do they infect the host cells. So this is a virus. It's a tiny and disease-causing infectious agent. It's about size of 100 nanometer, and some people, this is kind of a half in between alive or non-live because it can only replicate within living cells of a host. But if it's outside of the living thing, then it doesn't do anything. And the virus, interestingly, it consists of a genetic material. It could be DNA or RNA. We call DNA virus or RNA virus. And that's surrounded by a glycoprotein coat, and that we call a capsid. So especially many, the common cold viruses are RNA virus. Remember, RNA as a single strand RNA is more susceptible for mutation. So that's why every year we need to have a new vaccination for common cold. So let's take a look how the virus infect our body. So this is a diagram, and I put these steps for the virus infect cells with a host within a host organism and causing disease. So the first, virus will enter into the host cell. You can see it attach and endocytose. And second, disassembly of virus to release their genetic material. So once it's inside the cell, it will disassemble and it will release inside genetic material. It could be RNA virus, RNA, or it could be DNA. Very interestingly, if it's RNA, that will be inside the ribosome. It will undergo multiple RNAs, and it will make a protein synthesis. Or another way of DNA it can also integrate its RNA, converting into DNA and input into the genome of the host cell to use the machinery of the host cells translation and generating protein synthesis. And that is requires of converting RNA to DNA, which is counterintuitive because we, from the central dogma of molecular biology, we know that It's always information coming from DNA to RNA to protein. Now we are talking about this. Actually, this discovery is happened in the HIV virus. HIV virus is an RNA virus. But somehow they put their RNA into DNA of the host genome, which is only possible by converting RNA into DNA. And so people figured out this virus actually contains reverse transcriptase, which means causing a reverse transcription or RNA into DNA so that the virus can integrate into host genome. So that actually led to Nobel Prize, and the discovery was by Howard Hamin and David Baltimore, who later become a president of Caltech. So then they replicate the viral genetic material. You can see the replication of multiple RNAs and translation of the viral genes to produce new viral proteins. And then they will assemble new viral units, and these multiple virus particles will now release from the host cells. So these multiple infection and generating and release of multiple virus particles, these part of mechanisms are very important, because when we are coming up with a treatment to stop infection, we must know of these mechanisms, and we may want to find out which part we have to block it.